Hi everybody, welcome back. I'm changing things up a little bit. I'm using my tablet instead of my desktop to record on so that I can more easily write and be able to show you things. So today we are talking about wonderful, awesome water. As you know, water is H2O. And basically what we're talking about today is the physical and chemical characteristics of water, as well as a little bit of acid-base chemistry, which hopefully is a review. And we'll also talk about buffers, which is how the body maintains certain pHs. So here are our objectives listed for you. Again, you can find this presentation on Moodle or Facebook so that you can print out these objectives and then work on them afterwards. So when we talk about water, we need to talk about, how important water is. The textbook says that water is central to biochemistry for three major reasons. One, almost all biological molecules assume their shape due to water. Also, the medium for the majority of biochemical reactions is water, and water participates in many biochemical reactions, such as hydrolysis, where you use water to break a bond. Now, let's get into the physical properties of water. So, a water molecule consists of two hydrogen atoms bound to an oxygen atom. The hydrogen bond, or the hydrogen-oxygen bond distance here is 0.985 angstroms. And your bond angle between the hydrogen, oxygen is 104.5. Now, what's interesting is oxygen does happen to be a tetrahedral molecule because you have lone pair of electrons here. And if you remember, or if you don't remember, you can go to the Wikipedia page and review on tetrahedral molecular geometry. Let's say if we take methane, right, which is tetrahedral as well, the bond angle between the hydrogen-carbon-hydrogen bond for tetrahedral molecules is actually 109.5. Now, the bond angle in water, being 104.5, is actually smaller than your typical tetrahedral bond angle because of your lone pair of electrons here on oxygen, pushing the hydrogens closer together, taking up more space on the oxygen than they would if there was actually a covalent bond here. Now, because of this, we actually get a partial negative charge on the oxygen with a partial positive charge on our hydrogen, creating a dipole moment. And this dipole moment is really important because it's what can allow water to hydrogen bond. Now, a hydrogen bond is where a hydrogen is bound to an electronegative atom, such as oxygen, that interacts with another electronegative atom, such as oxygen. So as you can see here in this table, we have a variety of different hydrogen bonds that can occur between our hydrogen bond acceptor, which is our electronegative atom, and our hydrogen bond donor, which is the atom that contains the hydrogen. And we get a variety of these different types of bonds occurring in proteins, as well as DNA or RNA, and in water. So as you can see here in water, our hydrogen bond distance, which is the distance between the hydrogen and the oxygen, is 1.8 angstroms. Now, hydrogen bonds, all by themselves, may not be very strong. One individual hydrogen bond is only about 20 kilojoules per mole. Compare that to a covalent bond, such as here, which is 460 kilojoules per mole. However, if you get enough hydrogen bonds going on between molecules, that 20 kilojoules per mole per hydrogen bond can add up really quickly. For instance, when you have hydrogen bonding in base pairs between, say, G and cytosine, you have 3 hydrogen bonds between guanine and cytosine in the DNA. So that's 20, 40, 60 kilojoules per mole per base pair. So for every GC base pair, you're getting on average 60 kilojoules per mole of stabilizing energy in that bond. Now, remember what I said, so the more hydrogen bonds you have, the more stable your structure is. And indeed, if we look at ice, we can see that water plays some interesting properties in ice. So ice, the molecules of water in ice form cage-like structures, as you can see here, by orienting themselves in order to optimize the number of hydrogen bonds formed. So in ice, you get the most hydrogen bonds that a water molecule can form, which is 4 hydrogen bonds per molecule. And what that does is, because of this cage-like structure that occurs, ice is less dense than liquid water. It is actually 0.92 grams per mole, where liquid water is 1.0 grams per mole. And this is what allows ice to float on top of the water. The book points out that this is actually really important. If ice did not float, if ice sank to the bottom, you could picture a lake, right? And if the ice sank to the bottom of the lake, down here, where it's really, really cold, you have very low temperatures, the ice may never melt. And eventually, it would get colder up, and then you end up with a big solid lake. Instead, since ice does float, eventually when spring comes around and the temperature increases as the sun melts that ice, you then get, your liquid lake back. But I am digressing. So let's get back to the topic at hand. So as water melts from ice into liquid, these hydrogen bonds become much more dynamic. So they're still forming in the water molecules, but they're fluctuating. So they'll form, they'll break. They'll form, they'll break, all the while the water is moving throughout the solution. These molecules are moving. So you can get a number of different ring structures in hydrogen bonding in water. Because we can't really analyze this directly, this is all theoretical predictions based on spectroscopic data. But you can get the idea. The hydrogen bonds are still forming in water, but they are fluctuating. But even the book does say that because these hydrogen bonds occur, it actually makes water a pretty interesting solution when we compare it to methane. So water itself has a very high boiling point, 100 degrees C, which is significantly higher compared to other molecules with similar molecular masses that have significantly lower melting temperatures. And this is because of these hydrogen bonding interactions. So we have some other non-covalent interactions, such as, ionic interactions. So these are formal charges interacting with each other. Dipole interactions, where you get non-formal charges, but you get some dipoles occurring, but yet these aren't, necessarily hydrogen bonds. Van der Waals interactions, which are weak interactions between all atoms regardless of the polarity. These arise from electrostatic interactions among permanent or induced dipoles. And if we think about that, the hydrogen bond is a special kind of dipolar interaction. And then we can also have the hydrophobic effect, which is where hydrophobic molecules interact with each other in order to shield themselves from the solvating water molecules in the solution. Here we can see examples of types of non-covalent interactions in biomolecules. So we have our hydrogen bonds, our ionic interactions that can occur in functional groups such as the amino acids and proteins, as well as the hydrophobic interactions. Here we can see that these two hydrophobic groups aggregated in order to shield the inner surface from water. So, if we're talking about the hydrophobic effect, we need to talk about hydrophobic molecules. So hydrophilic molecules are those molecules that have polar and ionic materials that easily dissolve in water, or they're water-loving. So here we see our salt. Here we can have some table salt, sodium chloride, and we dissolve it in water. And it's readily dissolvable because what happens is the positive hydrogens on oxygen will interact with the negatively charged chloride ion and shield it from the sodium, and the sodium actually gets surrounded with the oxygens on the water oriented themselves to shield that structure. Now, this solvation sphere is actually going to be larger than, say, one layer. As you can see here, we get multiple layers of waters orienting themselves to shield the charges of the sodium, the chloride from each other, readily dissolving the material in the water. Now, hydrophobic molecules are nonpolar and insoluble in water, and they're actually water-fearing. Is hydrophobic, water-fearing. So what happens is when you try to dissolve hydrophobic molecules in water, the water actually has to orient itself and solvate the nonpolar solute in such a way that the waters will try to form hydrogen bonds with itself. And this is actually both entropically unfavorable, so we have a decrease in entropy, as well as enthalpically unfavorable, because you're trying to dissolve a non-polar solute in the water. So sort of like with oil in water, what you see is when you dissolve particles of nonpolar or hydrophobic molecules in the water, instead of having single particles, they'll actually aggregate into one whole particle. And this is actually what we call as our hydrophobic effect. So now, instead of the water molecules having to solvate each particle on its own, by the particles aggregating, we can release some of these bound waters at the aggregates interfaces, and the water now only has to solvate a smaller surface area of the aggregate. So now we've increased the entropy because we've released some of these bound waters back into the bulk solution that were at these interfaces. And we've also increased our entropy because now we have favorable van der Waals interactions occurring at the aggregate's surface, or in the interior of the aggregate, so to speak. Now, amphiphiles are molecules that contain both hydrophobic and hydrophilic segments. A number of biomolecules, such as lipids, proteins, and DNAs, are amphiphiles, in that they contain, for instance, in lipids, we can have a carboxylic acid head group that's hydrophilic with a hydrophobic fatty acid tail. That's just, a chain of carbon hydrogens. So what happens is the hydrophilic head, will have favorable interactions with the water molecules, forming favorable hydrogen bonding. However, the tail being hydrophobic, we have unfavorable caging of the water along the tail. So in order to minimize this, biomolecules will tend to aggregate, as we can see here, so that we get favorable hydrophobic interactions between the tails, favorable solvent interactions at the head group, so we get favorable hydrogen bonding here. And then we only have unfavorable hydrophobic water interactions at the outer edge of the tails. Now we can, molecules such as lipids will further decrease these unfavorable interactions by forming micelles. So now the entire outside of the micelle is formed by these hydrophilic head groups, forming favorable interactions and hydrogen bonding with water molecules. And now all of our hydrophobic groups are sequestered in the inside of the micelle, so that you have favorable van der Waals interactions between the nonpolar groups. So this is along the lines of the parable that like dissolves like. Okay, so nonpolar solutes like to be with nonpolar solutes, and then polar solutes like to be with polar solvents. And this is actually the driving force of a lot of folding in protein as well as DNA, in that hydrophobic regions in the protein will sequester inside the protein, while the protein will have its hydrophilic groups on the outside interacting with the water molecules. So this is what helps drive tertiary structure a lot of the times. Again, with DNA, when you have a double strand of DNA, you have a hydrophilic backbone, which is your sugar phosphate backbone, and then you have your hydrophobic nucleotides, which are mostly aromatic rings, which, aromatic rings are nonpolar, and so those are in the inside of the helix, shielded from water. Now, in addition to micelles, we can actually form lipid bilayers as well. Here, you have a layer of your hydrophilic head group. In the middle, you have your hydrophobic tails, and then you have another hydrophilic head group on the inside of the bilayer. And these are actually the basis of the plasma membrane, right? Again, all of this is driven because the biological matrix that these molecules are in is all water-based. So, when we're talking about water, and then we're talking about plasma membranes, which are, the outer membranes of the cell, we need to talk about osmosis and diffusion. So osmosis is actually a type of diffusion. When water is diffusing in and out of membranes, we call that osmosis. So in this instance, osmosis is when water flows from a region of high water concentration, such as our beaker, into a region of low water concentration, such as the tube containing, we'll say this is salt water, okay? So we'll say inside our tube is a high concentration of salt water. Now in this instance, our tube is semi-permeable to only water. So in order to get the same concentration of sodium chloride on the inside of the tube as it is on the outside of the beaker, water is going to flow in to the tube. Now you do realize that we will never get equal because there's never going to be any sodium chloride here in this beaker. So in order to stop the flow of water into the tube, we have to use osmotic pressure. So osmotic pressure is the pressure required to stop the inward flow of water from the beaker into the tube. And this is how cells maintain their concentrations of salts inside the cell, as well as the osmotic pressure in and outside of the cell. Now, dialysis is a biochemical technique often used to exchange or purify samples. In dialysis, we have a dialysis membrane or a bag that contains small pores. Now, based on these pore size, certain molecules can get through. So molecules smaller than the pore can diffuse in and out of the bag, where molecules that are larger than the pore cannot. So in this instance, as you can see, the small molecules can flow freely in and out of the bag. If we want to get rid of all these red dots, we'll call all of these red dots sodium. Let's say we want to get rid of the sodium. What we'll do is we'll remove the water after dialysis, add in more fresh water, and then reallow the dialysis to occur. And then we'll repeat that, I don't know, maybe 5 times till we get to essentially no sodium. We'll never completely get rid of all the sodium just because of the nature of equilibrium, right? You're always going to have something there. So the reason I wanted to talk about osmosis and diffusion is our cell membranes are permeable to only water, which means only water can flow freely in and out of the cell. Now, when we have the same amount of water coming into the cell as we have coming out of the cell, we are isotonic, which is good. This is our happy cell area. However, if we have more water coming into the cell than we do having coming out of the cell, now we're hypotonic, and eventually our cells can swell and burst. Now this can occur when we have very low salt, say in our bloodstream, and our red blood cells have much higher salt, so water is now going to flow into the cell in order to try to equilibrate that salt concentration. Now the opposite can happen. If we have more water flowing outside of our cell than we have inside of their cell, we have a hypertonic, and this is going to dehydrate our cells. This occurs when we have too high of salt outside of our cells than inside of the cells. So let's say you eat too much salt in your bloodstream, and now your red blood cells are going to shrivel up, and you'll actually get dehydrated. Both of these pictures here are essentially the same. It's just demonstrated a little differently. Here we can see the particles versus the water flow, and I just wanted to kind of give y'all 2 different perspectives. All right, so that pretty sums up, we've talked about the physical characteristics, we've talked about osmosis, and now we're going to talk about how water is actually a weak acid. So believe it or not, liquid water actually does dissociate into hydronium ions and hydroxyl ions or hydroxide ions. And we can measure this equilibrium, and I can go through how it's derived, but basically pure water, we have 1 times 10 to the negative 7th moles of hydronium and hydroxyl, which if you take that into the pH log is going to be a pH of 7. All right? So we can actually change this pH by adding more acid or adding more base, which will push our equilibrium of our water either one way or the other. And it actually sort of makes water a buffer. Not that water is a very good buffer. However, let's look at the pH of some other liquids. So very acidic, very strong acids. We have HCl. We can also have gastric juice, lemon juice, black coffee, slightly less acidic. We can also look at very strong bases such as sodium hydroxide, ammonia, or bleach. And then if we look around 7 or around neutral, we have milk and saliva, human blood, tears, seawater, and egg white. Now, the majority of biological fluids, such as blood and tear, likes to maintain a pH of 7. Our bodies want to maintain these pH because a lot of time this is the optimal pH for enzymes to react and catalyze reactions. However, when we have reactions occurring, such as A plus B yields C plus D, C may be our product that we carry on into another reaction. Well, D may be a metabolite of some sort or a byproduct that is acidic. And our biological fluids, or the pH inside the cell or in the blood, wants to maintain a pH of 7. Well, if you have reactions occurring that are producing acidic byproducts, how do these fluids maintain that pH of a 7? And they do so by utilizing buffers. So let's talk about buffers. So buffers are mixtures of weak acids and their conjugate bases. So some basics. Buffers resist change in the pH. At a pH that is equal to the pKa, there's a 50-50 mixture of acid and anion forms in the compound. The buffering capacity of an acid anion system is greatest where the pH is equal to the pKa, and the buffering capacity is lost when we go above 1 or less than 1 pH unit from the pKa. So let's put this in a picture form. So acetic acid is actually a weak acid, which means it can act as a buffer. So here we have acetic acid and its conjugate buffer acetate. So say for instance we have a mixture that's equal 50-50, 50% acetic acid, 50% acetate. If we add acid to our solution, we'll shift our equilibrium over to our acetic acid compound, which will then shift back to our conjugate base, releasing water, neutralizing that added acid. We can also do the vice versa. If we add base, we can neutralize that base using our weak acid to make our conjugate base and releasing water. As we can see here in this graph, this is a titration curve. So I don't know if most of y'all should have seen titration curves already, but here we have the plot of pH versus added equivalents of base. So as we can see, as we add base and we approach the pKa of our acid, we see that our slope gets very low, that we're not seeing a lot of change in the pH as we add base. So from 0.2 to about 0.9 equivalents of base, we see very little change in our pH. However, as we get to very high base added to the solution, eventually we use up all of our conjugate acid in solution, and now all we have is conjugate base in solution and excess base, meaning that our pH goes up and is no longer controlled. But as long as we have some conjugate acid in solution here in this range, we can neutralize some of that added base, which makes this a really good buffer around 4.76. So let's say, what if you want to buffer a different pH? Well, there's other weak acids we can look at. For instance, we can look at ammonia if we want to buffer around 9.25. We can look at phosphate if we want to buffer around 6.8. And again, if we want to buffer around 4.7, then we would look at acetic acid. Remember, you want to choose a buffer that's within 1 pH unit of the pKa. So really, acetic acid would be good for any pH between 3.76 and 5.76. Phosphate is good between 5.86 and 7.86, and so on and so forth. So let's say we want to make a buffer, or we want to determine how much of the conjugate acid or the conjugate base is in solution, or if we want to predict the pH. We'll do that utilizing the Henderson-Hasselbalch equation. This is an equation that biochemists use all the time. Here we can see our chemical equilibrium, where our weak acid is in equilibrium with hydronium and our conjugate base. And if here we have our equilibrium constant, where Ka is equal to H plus A minus over HA. If we rearrange our equation to solve for H plus, we get this. Again, if we put it to our pH scale, the negative log of the hydronium cation is equal to the negative log Ka minus the log of the conjugate base over the, or of the acid over the conjugate base. We then can convert this into pH because pH is equal to negative log of H. So we have the pH is equal to pKa plus log of A minus over HA. Now remember, I told you when you're 50% conjugate base, 50% acid, we have the pH is equal to the pKa. Now that's because if you're equal in concentration to your conjugate base over your weak acid, this becomes 1, where the log of 1 is equal to 0. Therefore, when these two concentrations are equal, this value falls out as it becomes 0, and now your pH is equal to your pKa. Now we also can have polyprotic acids. So these are acids that can dissociate more than one proton. And what you get is you get a titration curve with 3 plateaus, say, for phosphoric acid, since it has 3 hydrogens that can dissociate. So your first hydrogen is going to dissociate around 2, your second hydrogen around 8, and your third hydrogen around 12. So you can actually use phosphate to buffer 3 different pHs based on which hydrogen species that you start out with. So let's do an example of buffer. So a lot of the buffering in the blood is actually through phosphate buffer, since it's really good, as you can see, around 7 to 8. However, bicarbonate buffers are used a lot in the blood as well. So as you can see, here is our chemical equation for the bicarbonate buffer. So you can have carbon dioxide plus water. It's going to use bicarbonate, and then you can get bicarb, the metabolic bicarb. And what can actually happen is through respiratory control or the concentration of CO2, we can actually maintain the pH in the blood. So the optimum pH in the blood is 7.35 to 7.45, which is actually the pKa of this reaction here. So if we actually increase our CO2 levels, we can actually pull the reaction closer to the more acidic pH, which is pH is at 7.34. So if that happens, if we get too acidic, we can increase our breathing rate in order to pull more CO2 out of the blood, which will then push the reaction over to the more basic pKa side, or this side of our reaction, which is the HCO3. Now, if the blood gets too basic or we verge on alkalosis, we can slow our breathing down or actually cause hyperventilation at the same time too, to increase the CO2 levels in our blood. And so, again, we can control the pH through our respiratory rates because of the either presence or removal of CO2 in this bicarbonate buffer. It's just something cool. I think there's a blurb about it in your textbook. If I didn't really describe it very well, you can take some moment to read about it further. So I really think that's sort of the end of our lecture for today. And I'm not going to keep those annotations. So, again, if you have any questions, please feel free to post on the Facebook page or email me or stop by my office. And if things weren't clear, we'll try to make them clear. Read the textbook. And I think in class, I have actually a really cool activity that we're going to do where we look at aspirin overdoses and how that affects the blood buffering, the bicarbonate blood buffering to maintain the pH. So that will be a fun way for us to explore further buffers, pH, as well as linking into the medical side of things by looking at what happens to the pH of our blood under an aspirin overdose and how we can fix that. All right, so y'all have a great day, and I will see you in class.